I have the pleasure of introducing Dr. Nikolai Petrovsky, who is Research Director of Vaccine, a biotechnology company focused on vaccine development. He is a clinical endocrinologist and has a PhD in immunology. His work in vaccines and artificial intelligence has been awarded over $50 million in grants from the United States National Institutes of Health. Dr. Petrovsky will present on the origins of COVID-19 based upon computer modeling and vaccine design. I will turn the floor over now to Dr. Petrovsky, and thank you for presenting.
So thanks, Kevin, for inviting me to give a talk, to go delve into conspiracy theories. And as you know, there have been some big players involved in this, and there's been global politics involved in this. There's been very little science actually in the debate about the origins. You've had Tony Fauci on one side, you've had the Chinese government aligned on the other. The Chinese scientists are gagged largely, so you can't hear from them, apart from a few who are allowed to talk. You know, you had obviously Donald Trump and Biden weighing in, and then a whole lot of intelligence services, but very little science. And as I'll say, when the scientists did get involved, it turned out that those scientists themselves were creating conspiracies. So how did we get into this? We're vaccine developers. So how did a vaccine group get dragged into, you know, this highly politicized question of where did the virus come from? And the answer is, you know, it was all quite innocent and unintentional. We, as I say, took the genomic sequence back in January 2020, way before WHO called it a pandemic. We were arguing publicly that this clearly was a serious virus, and we needed to start working out two things, like how to protect against it and where did it come from. So we took the genomic sequence, you know, we tried to work out what this virus might behave like by blasting it, and obviously came up that it had about 80% genetic homology to the SARS virus, which we'd worked with before. So we said, great, that gives us a good lead. And we started our vaccine program. But there were a few things that just really worried us from the first time we looked at the sequence. Because when we're comparing it to SARS, we can see, yes, it's 80% similar, but there were then these glaring differences. And one of those glaring differences that just stood out was the furin cleavage site, which was not in SARS, but it was in SARS-CoV-2, wasn't in any related viruses between SARS and SARS-CoV-2. And, you know, so there's a lot of coronaviruses that are in that broader family. So where did a furin cleavage site come from? Because again, viruses can only either create something de novo through mutations, or they acquire it by recombining with other viruses. And I'll go into a little bit later, neither of those were plausible as where the furin cleavage site came from. It was 4 amino acids long. So if a virus is acquiring a new feature through mutation, it mutates 1 amino acid at a time. It can't do multiple. That the combinational sort of issue is too large. So a virus will mutate one. But how does it know to mutate 4 residues side by side sequentially to create a furin cleavage site? Because it doesn't know where it's going. It's doing this randomly. So it can't say, I need a furin cleavage site. I'm just going to progressively mutate this region to find a furin cleavage site. That's just not conceivable. So it can't occur through mutation. It can only occur through recombination, which means there has to be an extremely closely related coronavirus that has that furin cleavage site, identical furin cleavage site, and it actually swaps that piece of genetic material into the SARS-CoV-2. But there was no such known coronavirus out there that was closely related that had a furin cleavage site that could do that swap. So this is inexplicable. This is weird. When you look at either side of the furin cleavage site and compare it to the SARS site, they're identical on each side of the furin cleavage site. So it's like 100% homology just in that short region. And we're going, well, and in fact, for our vaccine, we said, well, we have to get rid of this furin cleavage site. It's clearly not natural. It will cause instability. And we decided we'll just remove it, take all those, revert it to the SARS sequence. And that's what our vaccine has in it. And the vaccines work brilliantly, which says that furin cleavage site doesn't have to be in the virus, and it may not be natural to the virus, which still leaves this question then, where the hell did it come from? And how did it get in there? And again, we can't answer that question. We can speculate, but it was just like, that was, blindingly obvious to us from the first analysis. This is really weird. And the more we looked, the more weird features we found. Now, nature can throw up weird features. So I'd be the first to admit, nature can do anything. There is no, nature controls everything and can create things that we can't even conceive of. So you can't say this couldn't have happened somehow naturally, but it just was unusual. So we then took out the furin cleavage site to make our vaccine because we didn't believe it should be there for whatever reason, and that worked. And so that showed we could make a stable protein. We then went about modeling the spike protein structure, because to get a structure, you have to purify the protein, crystallize it. It's a laborious process. So we do a lot of in silico using artificial intelligence to model proteins from genomic sequence. So we made a model of the spike protein, and we were able to identify human ACE2 as the receptor, because we modeled human ACE2 and we showed the two were binding tightly, more tightly than even SARS bound to human ACE2, which said, this virus is even better suited to binding the human receptor than the original SARS virus. And again, as we did this, the Chinese were similarly publishing data. They had the virus, so they were publishing the physical data, basically almost simultaneously with us, because we didn't have the virus, actually predicting all of these things from our modeling approach, which said our modeling was good because, again, it was correlating with the data the Chinese scientists were publishing at the time. So what was not known at the time, and what was speculated, and what we speculated, is that, like the original SARS, this virus came through some intermediary host. So the way that SARS got into humans, and this was nicely eventually mapped out, is that originally it was a bat coronavirus. That bat coronavirus got transmitted to civet cats, and those civet cats then transmitted it to humans. And the reason it had to use that route to get to humans is that the bat virus doesn't infect humans. So that's the original SARS. So it had to mutate enough to get into a civet cat, to mutate to adapt to civet ACE2. And then from a civet, it was able to just jump across to humans, and then it rapidly acquired new mutations that made it actually able to bind human ACE2 better. And that's when it became an outbreak and started to spread rapidly through humans. So this was beautifully mapped for SARS. So the obvious thing to think when we saw COVID is, okay, so it must have come the same route. It must have come from a bat, jumped to some intermediate host like a civet cat or something else, and then jumped across to humans, probably, either on a farm or wet market, because you've got all of these wild and farmed animals all in very unhygienic conditions, mingling with people in China, where this outbreak started. So we thought that, like everyone else, that was the hypothesis. And we thought, well, we've built this beautiful computer modeling of the spike protein and human ACE2, and it's working beautifully. It predicted the receptor. Could we model all of the ACE2s of all these exotic species for which there's no structures, so no one had any data on them, except the genomic sequence? We now have genomes on pretty well most animals on the planet. There is a published genome. So we thought, let's search in all these species genomes, find their ACE2 receptor by homology, because it will have some homology to the human and the known ACE2, and then we'll model them all, and then we'll dock the virus onto all these ACE2 to find the one that has the best fit, because that's where it must have come from, because if it's come from an animal, it will be adapted for that animal's ACE2. So very simple, straightforward hypothesis. So this is just some data looking what we did right in those first few days in January 2020 when we were trying to understand what the hell this virus was about. So we were comparing the genomic sequence of the SARS spike, the original SARS, with the COVID-19 spike. You know, as I mentioned, there's various areas where the sequences are identical. They're pieces, often just single amino acids, where you see differences. And so, again, this is quite normal. But what was really evident to us is that there were regions that were more similar than you might imagine, but certainly the SARS sequence didn't have a furin cleavage site. So this is just the model that we built of the original spike protein that we then used in this study that I'm talking about. And obviously, having identified ACE2 as a receptor, we were then able to say, can we model ACE2 from the different species, dock the virus, and then work out what species the virus actually came from. So you can use these models, as I say, for lots of purposes. So this wasn't the only question we were addressing with the model, and it wasn't actually the core question because it was really a side thing. We just thought that was interesting to do as scientists. So yeah, and we'll get into this, it gets a lot more complicated, but over that first few months, there were some interesting developments. So there was an announcement from China in the media that they'd found COVID-19 in a wild animal, which was a pangolin, which, you know, most people aren't even sure what a pangolin is, but it's like an anteater. These are almost on the verge of extinction, and in fact, it's illegal in China to traffic a pangolin. So it turned out that the whole story was almost a hoax. They were publishing data on a pangolin coronavirus that had been identified a number of years before, which actually wasn't that similar to COVID at all. It was almost like a grab for media attention by a group of scientists who didn't quite know what they were doing. But, you know, for a while there, everyone said, oh, we now know where COVID came from. It came from pangolins. And it's been hard to eradicate that whole story, even though it was all proved to be false and incorrect. And you can see that if you compare the pangolin sequence to SARS-CoV-2, you can see that there's similarity, but in the spike protein, and is concentrated in this area. But if you go to the whole, and this is why they claimed that this was the source of COVID, was just if you look at the spike protein, it looks extremely similar. But if you actually look at the whole virus, you know, in the pangolin and the whole virus of COVID-19, they're incredibly dissimilar. So to claim that this was the source of the COVID-19 was not true. What was interesting is the pangolin virus could be the source of the spike protein in COVID-19 because of this similarity. But that would imply that someone had moved the spike protein from a pangolin into another coronavirus. The other thing is the pangolin coronavirus didn't have the furin cleavage site. So if someone had moved the pangolin sequence into some other coronavirus backbone to create COVID-19, they must have also then secondarily gone in and put a furin cleavage site in. And that sequence of events is interesting. And I'll come back to it because we can go to the DARPA grant that was put in by Eco Health Alliance a number of years before the pandemic, where in fact they were proposing to do exactly that, to move spike proteins from one virus to another and then secondarily to insert furin cleavage sites. So it's just interesting that the pangolin may not itself, the animal may not be responsible for COVID-19, but part of that pangolin virus may actually explain the spike protein in SARS-CoV-2. The other thing that happened around that time was that the Chinese from Wuhan Institute of Virology said they'd found in their freezer a virus, rat G13, which had the highest level of homology to SARS-CoV-2. And you can see here, if you compare rat G13 spike protein with SARS-CoV-2, you can see that it's almost identical. There's an occasional, so shown here in blue, you can see there's a substitution in this amino acid. There's a few substitutions here, but you can see there's whole sections which are almost identical. And that was interesting for a number of reasons. People then said, well, did the Chinese make SARS-CoV-2 out of rat G13? Because, it wouldn't have taken much manipulation, obviously, to turn one of those viruses into another. In fact, there's about 40 to 50 years of evolution separate between the two viruses. But of course, in the lab, you can accelerate that. Viruses mutate very rapidly when you're adapting them to lab culture conditions. But overall, I think the overall consensus is no, that rat G13 is not the virus or the direct ancestor of COVID-19. But then the story got fascinating because when the Chinese didn't reveal where rat G13 came from in their paper in 2020 when they published it, it was then revealed, in fact, that this virus had been identified in a particular cave in a province of China that the Wuhan Institute of Virology regularly visits to sample viruses. Because a number of miners who were getting bat poo out of that mine all became sick, and 3 out of 6 died a number of years ago. And that brought the interest of the Wuhan Institute of Virology, because the Chinese scientists who looked after these patients concluded that they'd died from a coronavirus, and obviously it must have been a bat coronavirus. And why the Chinese, when they published rat G13 in 2020, never revealed where it had come from or the circumstances, in itself a mystery. Why didn't they want a connection made to where they actually found the virus? And that was revealed again by an American team called Drastic, who did all these searches and connections. And the Chinese eventually admitted, yes, that was true, but never explained why they hadn't revealed that. So rat G13 has a fascinating story even beyond the science. And as I say, it still remains the closest virus we have pretty well to COVID-19. All right, coming back to our mapping stories. So here is the relationship and homology map of all of the different species. This shows you, if you take the ACE2 genomic sequence from all of these different animals where there's a known genome sequence, you can map how related they are. So on this map, the distance of the line between two says how closely related the ACE2 in those two different species are. So for instance, human and gorilla, our ACE2 is pretty similar. Not surprisingly, because evolutionary-wise, we're pretty close to gorillas. Conversely, if you look at the similarity between human ACE2 and, for instance, wombat ACE2, something exotic, you can see that they're a massive genetic distance apart. So in other words, they're completely different. So what that tells you logically is that a coronavirus that infects a wombat because it binds wombat ACE2 is almost certainly not going to be infectious to humans, because the ACE2 is so different, it will be optimized for one, it won't cross over. So when you take human as the starting point and you first ask, well, where did the virus come from? The most likely place it comes from is a species where the receptor is closely related to the human. So here you can see rat and hamsters and rabbits aren't too far away from humans, so that could be a potential source. Maybe horse, camel, alpacas, it's a bit further, but still closer. When you go to, say, pangolin, for instance, you can see it's now a long way away, as is the civet cat and some of these. So this is already starting to sort of build a picture. And I'm trying to find bat here. There it is. So what you can see instantly is that bat ACE2 is an enormous genetic distance from human ACE2. And that explains why SARS had to jump from a bat to a civet to a human. In other words, it's jumping closer and closer. It can't make that one big leap. And so I can tell you definitively, COVID-19 did not come from a bat infecting a human. That scientifically, it's almost a ludicrous suggestion. And yet it continually was put about for the first few years. this could have been a wild bat that flew into Wuhan and infected a human. It's complete and utter nonsense. It's just, and we modeled this and we actually showed that COVID-19 doesn't bind bat ACE2. So it can't be a bat virus. It may have started off as a bat virus initially, but it would have had to have moved to a species closer to human. It couldn't have made the jump directly. So that immediately ruled out that whole story of this came directly from a bat. It didn't come from a bat. So if it came through a natural route, and again, we have no evidence of it coming through a natural route, it had to come through an intermediary species. So we just set about doing our simulations and looking at any species that was related to human ACE2. And you can do that. You can look at what are the residues in the spike protein that are critical to binding of the virus, which are shown here at these positions in the spike protein. So these are the key positions. So you can focus in on these and then say, well, what's the similarity? So if you look at these key residues that are mediating the binding between human and monkey, you see they're all identical. So although the ACE2 has differences between monkey and human, they're not in any of these key residues. Conversely, if you look, at say a mouse, you can see that only about half these residues are shared. By the time you get to a snake, which was again put up as a potential source of COVID, complete nonsense. None of these critical residues, or very few of them, are shared with humans. So that already says this is very implausible that it came from any of these animals at the bottom of this list, which includes the pangolin. So then, to make it more precise, we actually modeled these ACE2s, and I'm not going to go into all the complexity of what all these things mean, but essentially this is the quality of all our models. And so you can see they're all up 96 to 98. These are extremely robust models that we've built of all these different species of ACE2. So we end up with a structure. So this is a protein structure, and we're then able to use that structure to dock it with the virus to see how well they bind together. And so we end up with what we call a binding energy score. The more negative it is, the stronger the binding. So we've ranked them all here according to the predicted binding of the virus to these different species of ACE2. So you can see immediately, and this just came straight out of the raw data, here is the species that shows the strongest possible binding to the original virus, right at the top of the list. That was not what we were expecting. We're expecting humans to be somewhere down the middle, because this had only just come to humans. It shouldn't be adapted for humans. It should have some binding because it's clearly infecting humans, but it shouldn't have been optimized. So it shouldn't have been extremely high. And there should have been some other species to which it was adapted, because that's where it's come from, that should have been even higher. But what you can see is all the animals we tested, and we've subsequently done more, and the Chinese actually have similarly measured this physically and come to the same conclusion as have American scientists in the lab. No one can find an ACE2 that has stronger binding than the human ACE2, which would in itself say this virus was already evolved optimally to infect humans from what we believe to be the very first infections. And that's really challenging to understand. Again, a bit like the furin cleavage site, just could it be a complete coincidence, 1,000,000 to 1? Yeah, you can never exclude that. But how many 1 in a million coincidences can you find with a single virus before you say this cannot possibly be how this came about? So it's really adding all of those probabilities together that any one of these things could happen by chance, as I say. When you have a whole lot of things that shouldn't be happening by chance and you add them together, you have a really difficult time explaining what you're observing. In terms of the quality of the modelling, because it's the best modelling, we did it incredibly rigorously. We used a lot of supercomputer power donated by Oracle. It's good science. The models are flawless. This is a big surprise. This virus was pre-adapted to bind the human receptor, which we thought was a scientifically very important finding. Obviously, it might lead to other questions, but, it was a scientific observation that was robust. Here's the pangolin, poor old pangolin. As I say, we don't believe that the virus came from a pangolin. I don't think the Chinese seriously now believe that either. I think they've moved completely away from that. Apart from anything, there are very few pangolins in existence, which makes it highly improbable, you would think, that of a chance encounter between a pangolin and a human, because, as I say, they're almost extinct and they're heavily regulated. And you get a jail sentence in China if you're found trapping a pangolin or trading it for food or for its scales. A paper about surveys in Wuhan wet markets that was data from before the pandemic. Because this, again, if pangolins weren't in the wet markets in Wuhan, how could the virus have come from a pangolin if they weren't there? And in fact, this paper, who was surveying all the animals that were in the wet market, showed that there were illegal animals, but there were no pangolins, because, as I say, and no bats. And that's because, as I say, you go to jail for a long time in China if you're found with a pangolin. So not surprisingly, they weren't there. So there's no evidence this came from a pangolin. But there are some interesting perspectives that the virus from the pangolin, although it's not COVID-19, gives us about COVID-19, including the fact that its spike protein is remarkably similar, even though the rest of the virus isn't, suggesting someone might have taken that piece of the pangolin virus artificially and stitched it into some other coronavirus. So I think that's where the pangolin sits, but the pangolin itself is innocent. So just to acknowledge all of our great support that we've had over this last 3 years. That's my story, collapsing 3 years of crazy, intense work into an hour or so. And I'm happy to take any questions.
Well, thank you very much. And let me ask you back on proteins. It'll be a protein question right up your alley. Is that I had read, it was in the Bulletin of the Atomic Scientists, that when they looked at the codons, which codes for the amino acids, that those codons were not typical of, but more typical of another species. And so that we're not talking about the amino acid makeup that you looked at, but the other step of the codons that code for that amino acid, that those weren't lining up either.
Yeah, look, I've read those papers too, and I mean, it's intriguing. I'm not a codon expert. We do codon optimization to get our protein into insect cells, for instance, so we touch on it. But I think it is intriguing that again, people have done all sorts of interesting analyses from their perspective on this virus, and they find, again, unusual features. And any one of those you could say, well, again, maybe it's just chance. I mean, different codons appear different frequencies, but if it's slightly out of balance, does that mean anything or not? I mean, the furin cleavage site, there are two codons, one after the other, that encode part of that furin cleavage site, that those codons are almost never used by coronaviruses. And there are two of them together here, but they're commonly used by human experimenters when they're actually putting in genetic inserts. So they're very commonly used codons by humans. And so, again, I mean, it's layers upon layers of these features. And it's when you put all of those features together, any one of them can be dismissed. And that's what I'm trying to say. But when you see all of these put together, yes, probably some are just misinterpretations or just, variations. But how many of these unusual features do you need before you say, well, we need to ask questions how this could have happened?